Hi everyone, welcome back to Dento'ZEN. Today we are here with a new video where we are going to hear about amelogenesis. Amelo refers to enamel and genesis refers to formation. So amelogenesis is the process of enamel formation. In this there are two steps. In the first step there is formation of enamel matrix, which is mainly the organic component which mainly constitutes the enamel matrix proteins. And in the second step there is mineralization, that is addition of the inorganic material, that is minerals, to this organic matrix. So we can say that amelogenesis is the process of enamel formation, which is divided into two steps: enamel matrix formation and mineralization. What are the stages? Before that, let's know about the cell ameloblast, which forms enamel. It goes through various stages in its life cycle, and this is known as life cycle of ameloblast, in which there are 6 stages which we have already discussed in the last video. So please go through that video first so that you can understand amelogenesis in a better way. So in its first 2 stages, the cell prepares itself. In the next 2 stages, it actually forms enamel, and in the last 2 stages, it protects the enamel and helps in the eruption of the tooth. So during its third stage, that is formative stage, it forms a process which is known as Tom's process, which forms the enamel matrix, which is laid down in 3 layers. Along with that, there is immediate partial mineralization, that is 30% of mineralization occurs immediately. Mineral is added to this matrix, and after that cell takes a break, and then it carries out the final mineralization, that is full mineralization. So here two things are happening. First, the initial secretion of the enamel matrix along with the partial mineralization, which is called the secretory stage, and after a brief transitional period, cell will carry out the maturation of this partially mineralized enamel, and that is known as maturative stage. So before these 2 stages, some changes are undergoing to prepare the cells, that is known as pre-secretory stage. So stages in amelogenesis are 3, that is pre-secretory, secretory and maturative. Pre-secretory is further divided into morphogenic phase and differentiation phase when the cells are getting prepared. Secretory stage will involve the formation of Tom's process and how it helps in the formation of enamel matrix. Maturative stage will include brief transitional phase where the cells undergo changes, and the maturation proper where they will carry out the full-fledged mineralization. So 3 stages. So in this video we are going to cover amelogenesis in 4 parts. First part, introduction: what, when, where, how. Then stages, that is 3 stages: pre-secretory, secretory and maturative. Also we'll talk about enamel matrix proteins and mineralization key features. Then how amelogenesis is unique, and what can go wrong in the amelogenesis. For your short note, you can include stages, which is the most important part, but for your long question, you can write all the 4 parts. Let's begin. So before we start, I want you to quickly subscribe to Dento'ZEN. Also hit on the bell icon so that you remain notified about new video. Introduction. So what, when, where, how. You already know what is amelogenesis. Enamel formation occurs in 2 steps. When? It starts in the advanced bell stage of tooth development, when the enamel organ is bell shaped and its inner enamel epithelials, they change into ameloblast cell and start the enamel formation at the cusp tip. So it starts where it starts at the cusp tip and proceeds towards the cervical region. And how? Ameloblast cell will carry out all the stages of amelogenesis. Come to the second part, that is stages. There are 3 stages we already know: pre-secretory, secretory and maturative. In the pre-secretory stage, further divided into 2, morphogenic and differentiation. Secretory stage will involve the formation of Tom's process and how it helps in matrix formation, and the third stage, maturative stage, will discuss the brief transitional phase and the maturation proper. So let's talk about first pre-secretory, which has 2 phases: morphogenic phase and differentiation phase. So enamel formation begins in the bell stage when the inner enamel epithelial cells stop dividing and starts differentiating in the cuspal or incisal region first. So that means they will determine what type of tooth will form. So in the bell stage, because crown shape is determined, it is known as morphogenic. Morpho refers to the shape. At this phase, inner enamel epithelial cell has 2 ends. One end of the cell, which is towards the stratum intermedium, is called the proximal end of the cell, and the other end, which is towards the dental papilla, is called the distal end of the cell, as we can see here in this picture. Now, the enamel formation begins at the distal end of the cell. So let's see how these cells are there in the morphogenic phase. So they have 2 ends, that is proximal end and the distal end towards dental papilla. The cells are short, cuboidal or low columnar. Nucleus is oval, large, lying in the center. Golgi body and centrioles are in the proximal part, whereas mitochondria are dispersed. So the cells also have junctions in between them, as we can see here, which connect them, and these are known as terminal parts. Also, cells have a basal lamina which separates them from the underlying dental papilla cells, the blue line. Now, as the first layer of dentin is formed, cells get the signal and they differentiate. Now they become tall columnar. What are the dimensions of this cell? That can be your Viva question. So these cells are about 40 microns in length and 4 microns in diameter. Then, if we look at the organelles, nucleus now is in the proximal part, and other organelles have reversed. Golgi and central have come in the distal, and mitochondria seem accumulated in the proximal part. So, there is reversal of the functional polarity of the cell. Also, along with the proximal junctions, now there are additional distal second junctions. From this, it seems that actin-like filaments seem to be radiating out and form the webs. So we have two webs now, proximal webs, terminal web, and distal terminal web. What is the role of these junctions? These junctions, they hold the cells tightly and they will control what passes in between the cells. Now we come to the secretory stage, which starts with the formation of the Tom's process, and that helps in enamel matrix. Now this Tom's process forms at the distal end of the cell or the secretory end of the cell, and it divides the cell now into two parts, cell body or cell proper, and the second part is Tom's process itself. So it is partly separated from the body near the second junctional complex, distal junctional complex. But it itself is further divided into two parts. It has two parts, the proximal part, which is towards the cell body, and the distal part, which is towards the dentin. Now, key features, that is shape of this Tom's process, that can be your Viva question. Is this pyramidal or cone-like projection? And it is a blunt process. It contains secretory granules, rough endoplasmic reticulum, Golgi complex, and mitochondria, all the organelles of synthetic cell, because it helps in enamel secretion. With the enamel formation, this Tom's process will give an appearance between the junction of the cell and the forming enamel, which is known as sawtooth or picket fence appearance. So this junction in the ameloblast, between ameloblast cell and forming enamel, becomes sawtooth because of this Tom's process, because it is angulated at the cell body. So how it helps in enamel matrix formation? This Tom's process, when it forms first, it only has the proximal part, which lays down the enamel directly over the already formed dentin. Now, this first layer of enamel is rodless. It does not contain any prism, so it is aprismatic, and it gets immediately mineralized. Soon after this layer is formed, ameloblast cell will move away, it goes away, and now it will form its second part. That is, along with the proximal part, now it has the distal part, which is towards the forming enamel. And now the secretions, enamel secretion, becomes staggered. That means now it will occur from the two sides. What are the two sides? First side is the proximal part, which is close to the distal junctional complexes along the adjoining ameloblast. So it will secrete the first enamel, which is known as interrod enamel. So interrod enamel is secreted from the first side, that is the proximal part. So it will be secreted like this, if we see from the top, as a continuum along the periphery of the proximal part. And then it will create a pit. Between the two interrod, we can see a pit or a depression is created where lies the Tom's process, distal part of the Tom's process. Now this distal part of the Tom's process is the second side, and now it will form the enamel in this pit, which is known as rod enamel. So we can say that interrod forms first, followed by rod. So interrod is always step ahead. It is so because first interrod will form, it will create a pit in which the rod enamel will be deposited. Slowly and slowly, as it keeps on enlarging, it will put pressure on the Tom's process. Growing enamel will put pressure, and Tom's process will become narrow and will be squeezed out. So the space which is formed now will be filled with an organic material which is known as rod sheath. So here the rod sheath will be formed. So that is how interrod and rod enamel forms. And finally, when the Tom's process is lost, the ameloblast cell again becomes ovoid shape, and now again it has only one secretory site, and now the final layer which is deposited is again similar to the initial layer, that is rodless or aprismatic. So that is how a layer of enamel which contains rod and interrod, also known as prismatic enamel, is sandwiched between initial and final layers of rodless or aprismatic enamel. So we can see that one enamel rod is formed from two ameloblasts. According to another interpretation, enamel rod is keyhole shape and it has a body and a tail region. And it is said that one cell give rise to bulk of the body and other three ameloblasts will form its tail like this, as you can see here. So this keyhole shaped one enamel rod forms from four ameloblast cells. As we can see here, its head forming from one and its tail taking part from the four other ameloblasts. Also, one ameloblast can give rise to formation of four rods. How? It forms the bulk of the head of the one rod and the tails of the other three rods. So each ameloblast forms four enamel rods. Now, what are these enamel rods made up of? Enamel matrix proteins, they are amelogenins 90% and non-amelogenins 10%, ameloblastin, enamelin and tuflin. Now, next we enter the third stage, maturative stage, which is longer than the secretory stage, 3 times longer. That amelogenesis is very slow process, can take up to 5 years to complete in the crown of some of the permanente. To carry out maturative, ameloblast will take first break, transitional phase, and then will carry out maturation proper. So in transitional phase, it undergoes changes. It decreases in height, decreases in volume. Its organelles get autophagocytosed. The 50% cells die by apoptosis, but it will form atypical basal lamina. Atypical because it does not contain type 4 collagen, but it has other things like laminin, glycoconjugates, amylotin, and odam. So this basal lamina is attached to the ameloblast cells by hemidesmosomes, as we can see here. So this helps in the addition and also it controls the movement of particles. Now, after these changes, cell is ready. But what? It will first have to remove the matrix protein. Then only the space will be created to add mineral. So now the cell has to carry out two functions. Remove the proteins and add the minerals. So to carry out, the cell will now change its shape at the distal end. So when it has to remove the water and the proteins from the enamel, its distal end will become smooth. It is known as smooth-ended ameloblast. Its distal junctions will become leaky or loose, and these water and proteins will be removed from there. And when it has to add the minerals, it will again turn into ruffle bordered ameloblast. Distal end becomes ruffle. Irregularities will be there, through which the calcium will be added into the created spaces. So 80% of the time cell remains ruffle-ended and 20% of the time it changes to smooth-ended shape. So this cyclic change of the cell is known as modulation. So it can occur about 6 to 7 hours interval or 8-hour interval. So in a day we can see 3 modulation cycles. Now, how this process is carried out? So, extracellular enzymes, first, which are also released from ameloblasts, will break down the matrix proteins into smaller fragments, and these fragments are taken up by the distal leaky junctions of smooth-ended ameloblast. And when the space is created, the cell will change its shape, it will become ruffle-ended, and now it will add the minerals. Yes, the enzymes, that is enamel-lysine and kallikrein, which was previously known as enamel matrix serine protease, will need acidic environment. So acidification is associated here. To prevent that, ruffle bordered ameloblasts will now add bicarbonate ions, so that no reverse mineralization occurs. That means mineral whichever is added should not go back due to acidic environment. So to neutralize the pH, bicarbonate ions are secreted. The pH during the ruffle shape will remain acidic about 5.5, and as it becomes smooth-ended, pH becomes neutral. So ruffle-ended ameloblast shows considerable endocytotic activity. It has lysosomes, calcium binding proteins, and calcium ATPase activity, which helps in pumping the calcium ions into the enamel. Whereas smooth-ended has little endocytotic activity, it leaks small proteins, and it has no calcium ATPase activity. So it is mainly associated with removal of protein and water, whereas ruffle-ended is mainly associated with addition of calcium. Now we come to the ameloblast secretory products. So can secrete four things. First is the proteins of the enamel matrix, which can be of two types: amelogenins 90% and non-amelogenins, which can be 10%. So non-amelogenins can further be ameloblastin and enamelin and tuflin. Amelogenin key features: they are low molecular weight, they are located on X and Y chromosome, they can exist in 9 isoforms with differences in their amino acids, and they can be fragmented into smaller fragments like tyrosine-rich amelogenin polypeptide and leucine-rich amelogenin polypeptide by the slow processing of the enzymes. Ameloblastin was earlier known as amylin and sheathulin. Ameloblastin and enamelin undergo rapid processing by the enzymes, and they can be also broken down. So these are enamel matrix. They are located in matrix, whereas tuflin is found in the dendino-enamel junction. Now, what are their functions? Amelogenins form nanospheres, that is, between the crystals they will form nanospheres like this, so that there remains the space between the crystals. They do not fuse together during the secretory stage. Later on, they are removed as thixotrophic gels, and mineral is added in the maturative phase. So they helps in maintaining the space, they helps in the growth of the crystals. Ameloblastin and enamelin also helps in the nucleation and growth of crystals, and tuflin also helps in the nucleation of the crystals. The new protein amyloidin has been found in the basal lamina along with the other protein odum. It has said that it helps in the enamel mineral formation. Then certain proteins, transient proteins, are also liberated by ameloblast, which are dentin sialoprotein, dentin phosphoprotein, and they are found in the dentino-enamel junction. And also ameloblasts secrete the processing proteins, which are enzymes which breaks down these enamel proteins. Enamel lysine, which breaks down the amelogenins into small fragments, and kallikrein, which breaks down the remaining fragments, and the non-amelogenins. So it is a bulk digestive enzyme. So those are the four things which are secreted by ameloblast. Now we come to the key features of mineralization in enamel. It occurs in two stages: immediate partial mineralization, which occurs immediately as the matrix is secreted. So it occurs in the secretory stage. It is partial, so it accounts for 25 to 30%. So final mineralization occurs in the maturative stage, that is full mineralization. So immediate mineralization occurs in the matrix. How? There are no crystals, no matrix vesicles. So where it happens? So it takes the help of the crystallites of the dentin, which acts as the nucleating center over which the first crystals of enamel will be formed. So occurs as matrix is laid down, no matrix vesicles, no preformed crystals. Dentin crystallites act as nucleation, and initial mineral which is deposited is octacalcium phosphate, which is unstable, and later it breaks down into 2-hydroxyapatite crystals. Later on, final mineralization occurs by modulation in maturative stage, where the ruffle ended will add minerals and smooth ended will remove the proteins. So minerals which are added into the already present crystals, they help in their growth and the thickness. So crystal size increases from 1.5 to 25 microns, and their shape changes from original ribbon-shaped crystals to hexagonal or rhomboidal shaped crystals. Now this mineralization begins at the cuspal or incisal region and goes towards the cervical region. So if we see the progressive mineralization in the crown of a tooth, we'll see that it occurs in the two direction. That is, it is starting from the cuspal and going to the cervical region. Also, it is starting from the inner depth and going towards the surface. So that is the two directions: cuspal incisal to cervical and depth to the surface. Also, if we look at the advancing front of the mineralization, initially this advancing front is parallel to the dentino-enamel junction, and later on it becomes parallel to the outer surface of the enamel. Now, the rate of enamel formation is about 4 microns per day. So, according to that, about 1 millimeter of enamel thickness is formed in 240 days. Slow process, but the rate is more in permanent as compared to the deciduous stage. So, now we come to the third stage of this video, third part, that is how it is unique. Enamel formation, cell forming, is ameloblast cell, which is ectodermal in origin. For all other hard tissues, the cell is ectomesenchymal. Now the proteins are non-collagenous in enamel, that is amelogenins and non-amelogenins, whereas in all other hard tissues, collagen is the major protein or the principal protein, helps in mineralization. Now distinctive organic phase is not seen in amelogenesis because the matrix which is secreted gets immediately partially mineralized, whereas in all other hard tissues, unmineralized matrix is present, which later on gets mineralized. Associated matrix vesicles are absent, whereas they help in mineralization of other hard tissues, and they are present. So amelogenesis cannot happen later on because the cells undergo apoptosis, whereas in all other tissues they form throughout the life because cells are there. Now, what can be the defects in amelogenesis? They can happen if they can affect the cell ameloblast. So these can be infections, trauma, chemicals, and metabolic changes. If they occur during the secretory stage, so they can decrease the thickness of enamel, so it leads to enamel hypoplasia. Full thickness is not formed. And if the cell is affected during its mineralization stage, then it leads to a condition which is known as enamel hypomineralization. So now we have covered the enamel formation in all the four parts, that is introduction, stages in amelogenesis, how amelogenesis is unique, and defects. Now we come to the summary. We are summarizing the stages. First is pre-secretory stage. It is two parts: morphogenic phase, differentiation phase. Secretory stage, development of Tom's process and formation of enamel matrix. Maturative stage, brief transitional phase, and final mineralization, full mineralization, maturation proper. So that is stages in amelogenesis. Now let's check what have you learned. First, what are the three stages in amelogenesis? What is the enamel forming cell known as? Process at the distal end of ameloblast is called. Then enamel matrix proteins, their names. Which is step ahead in formation, rod or interrod? Cyclic changes in maturative state in ameloblast are known as. Percentage of immediate partial mineralization in enamel. Then which junctions are leaky in smooth-ended ameloblast? Is it proximal or distal? Then which hard tissue cannot form later in the life? And then what can be the defects in the amelogenesis? So that is all for this video, and good luck for your exams. If you really like the video, do tap on the like button. Also share the video with your friends. And keep watching, keep sharing, keep growing, keep learning. See you in the next video. Till then, take care. Bye bye.